Welcome to Stanford Legal, where we look at the cases, questions, controversies, and legal stories that affect us all every day. I'm Pam Carlin, along with Joe Bankman. Hi, Joe.
Hi, Pam. Pam, today we're going to talk about genes and DNA and how you might change your genes, your kids' genes, a mosquito's genes. What would you change, Pam, if you could change any of your genetic makeup?
Well, I would change whether I could fit into the genes I have. I suppose if there were a gene for patience, I would love to have that. How about you, Joe?
Well, it's easy for me, Pam. I would change the height gene because I'm vertically challenged, and my goal might be to be as tall as our guest today, Hank Greeley, who's Stanford Law School's Kate Edelman Johnson Professor of Law. I mangled that title a little bit. I hope you'll excuse me. Hank is long been one of the nation's leading authority on the ethics of medicine and science more generally and its intersection with law, director of our Center for Law and Biosciences. A lot of other great things in Hank's resume. Hank, welcome to the show.
Thanks so much. It's good to be back.
Yeah, we had a wonderful conversation with you before. So one of the acronyms that you hear a lot about these days is CRISPR. CRISPR then what, Hank?
Well, after CRISPR comes crispier and then crispiest. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which of course is why everybody calls it CRISPR.
I knew that, by the way.
Yeah.
Plus CRISPRs, you can verb CRISPR so easily. So you CRISPRed this or you're CRISPRing that. CRISPR is, I think, the most important bioscience discovery/invention of the 21st century so far, and maybe for many years to come.
How does it work?
It is a gene editor. It edits DNA, and although people worry about taking analogies and metaphors too seriously, this one really is almost like Microsoft Word's cut and replace function. What CRISPR will do is find a particular spot in your DNA. So let's say you're interested in a place in your DNA. DNA is made up of 4 letters, A's, C's, G's, and T's. There are 6.4 billion of those letters in your genome, in each of our genomes, which is like The Lord of the Rings plus The Hobbit 1,000 times over, literally. It's like all of F second, from F second 1 to F second 999. But you can say with CRISPR, find a spot that is AAA, CCC, TTT, GGG, ATC, and cut there. Cut that piece out. And CRISPR will go and cut that piece out.
I mean, literally cut it out?
Literally cut it out.
So where are they getting this DNA? Because they're not going through your whole body to cut it out.
So it depends. Some of the interesting possible uses of CRISPR are what are called in vivo CRISPR inside a living organism. So you could, in theory, go into the body, put the CRISPR into a virus or some other delivery tool, let it loose in your body and have it go into every cell it can reach and cut that DNA out. But there's more. Not only can you cut it out, you can attach to it another chunk of, I think I had cut out 20 base pairs, 20 letters. You can attach another 20-letter or 23-letter or 28-letter or 12-letter chunk and have it go in to the spot that you cut out. So, classic potential use for this. If this works, it'll be wonderful. The most common form of cystic fibrosis is caused by a change at the 508th base pair of a gene called CFTR. And most people have one reading, and most people with CF, with cystic fibrosis, have a different spelling there, which makes them sick and makes them die early. You could, in theory, take CRISPR, put it into, say, a virus for delivery, have somebody inhale it, it gets to their lungs, goes into the lung cells, cuts out the bad part, and replaces it with the proper spelling. It's a proofreader. And then your hope is it fixes it, the gene makes the protein, and their lung cells are healthy.
And by the way, I should say to our listeners, if you didn't get Hank's F second reference, F second contains a lot of legal decisions that would bore the pants off you were you to read them. much worse in reading, I think, Lord of the Rings 1,000 times.
Yeah, and the funny thing is with our students these days, they don't actually know that F2 was real literal books because it's just a computer file. It's just a name on a computer file to them. But it was 1,000, 1,200 page double column volumes. Kind of like DNA, though. Most of it is garbage. But there are some parts that are really important, and there are some parts that are really kind of beautiful.
So could you go in and like take your DNA, CRISPR it, put it in Joe, and then he would become you?
Well, you couldn't do it with all of my DNA. That's a lot. That's 6.4 billion base pairs. But let's say
For those of our listeners who are seeing this, let's say you just wanted him to have hair and a mustache like yours. Could we change his hair so it would be thicker and whiter?
That'd be scary. He'd be too good looking. I don't think we should do that. If we knew those genes, which we don't. I'll give you a better example. So most things we care about are actually genetically complicated, but there's a hair color that's actually quite simple. The really red hair, the freckles and red hair look, Scottish, Irish, etc., that's one mutation. We could, in theory, go into Joe's hair, which is not unhandsome, and change the cells that make the hair and change it from the gene that makes melanin, the dark pigment, into this red pigment version of melanin, and Joe, without Clairol or anything else, could end up growing red hair. If we changed his skin, we could also make him have freckles.
I'm into it. I'm game. I was just going to suggest that Clairol would be, I guess, an opponent of this, but as it gets better and better, we just change our genes?
Clairol will buy it.
So in kind of thinking about where this technology goes going forward, I mean, one of the things you've pointed to is when we know that there's a disease that has a genetic component, you could go in and swap out the genetic component that creates that disease. Are there other uses that we should be thinking about?
Sure. The big hope, though, is fixing diseases. I should say first, though, this is arguably invented in 2012 by a scientist at Berkeley named Jennifer Doudna and a European scientist named Emmanuel Charpentier. And then there's 7 other people who want to be among the 3 people who get the Nobel Prize for it. So there's a lot of fighting. It actually was invented about 3 billion years ago by anonymous bacteria. This was a way bacteria came up with to defend themselves from invading viruses. They'd chop up pieces of the virus and make the virus ineffective. So if you're a bacterium, you can use it to protect yourself. If you're humans, the main use would be to try to prevent or treat diseases. It's a better way of doing gene therapy. Gene therapy has been tried for 38 years, first approved by the FDA just last year. So the timing is good. CRISPR isn't the first way to edit DNA. That was actually invented here at Stanford by Paul Berg in the early 70s. But I like to analogize it to the Model T Ford. There were cars before Model Ts, but they were really expensive. They broke down all the time. You needed a full-time mechanic chauffeur, and you had to hope he wasn't a charming Irishman who would run away with your daughter, as in Downton Abbey.
Well, and if he had red hair, who knows what we could do with his genes?
Yeah, we need to work on his accent a little
I
bit.
think a combination of Ashkenazi Jew and red hair and freckles would be just irresistible.
Dynamic. Well, this is Stanford Legal, and today we're talking with our guest, Hank Greeley, about DNA and in particular about CRISPR. Joe?
And something came up in the news, Hank, about DNA and CRISPR involving an attempt, maybe successful, at using CRISPR at the embryonic level in China.
Right. This has eaten up most of my last 3 weeks. I've been buried in CRISPRed babies, which is kind of a bad image. A scientist in China named He Jiankui announced on the eve of a major international summit about human genome editing that 2 babies had been born whose genes he had edited when they were embryos. Now, I need to back up a little bit. We're talking about changing Joe and Joe's hair color. To do that, you just need to change the cells in Joe, and Joe, as an adult who may or may not, who's arguably competent, could give you informed consent for that. If we wanted to change all of Joe's genes, it would be really hard. He's got about 10 trillion cells that have human cells that have DNA in them. Changing all of those is going to be tricky unless we start when he's only got one cell. When he was a fertilized egg about 1 million years ago, he had one cell. If you change the DNA there, every cell that comes from that, which is all the cells he has now or has had in the previous trillion years, would be changed, including his sperm cells. So changing embryo cells changes all the cells in the body. It changes the cells in the body of a person who hasn't been around to give consent, and it changes their eggs or their sperm, which means, unlike changing Joe's hair cells aren't going to affect his kids. Joe's sperm cells would affect children. So this
Not the children he already has.
Not the ones he's already got.
Yeah.
But they call this germline gene editing because the eggs and the sperm are your germline. It passes down potentially to your children, their children, and their children. So what he did was change the germline of these babies. Not only was he experimenting on babies, but he was experimenting on babies in ways that could affect the babies' babies and grandbabies and great-grandbabies. This caused, for a variety of different reasons, a lot of outrage.
Well, one of the things, at least in the popular press accounts, which is what I read, is the claim was that he was making these babies resistant to HIV. Is that what
that was his claim? Now, I should preface all this by saying we don't know what the hell he did, if anything. No one has verified this. He has no other scientist we don't even know whether the babies exist. It would be a pretty bold fraud.
Would this be like Dolly the sheep?
Dolly the sheep did exist. We know that. But there was a Korean scientist named Wong Woo-suk who in 2004 claimed to have cloned human embryos. He just completely made it up and eventually went to prison. I think he did what he says he did in part because the results he presented are pretty bad. If he was really going to fake it, he should have given himself better results. So what he did, and this is part of the controversy, there's a gene in people called CCR5. It's not just in people, it's found in all mammals. One of the things it does is make a protein that sits on the surface of certain immune cells in your blood, T cells, part of the white blood system, part of your immune system. T cells are kind of famous because it's the part of the immune system that HIV likes to infect. And it appears that if you don't have any CCR5 proteins on your T cells, HIV either can't infect you or it finds it really hard to infect you. About 1% of Northern Europeans and non-trivial but smaller percentages of other people around the world have one of their two copies of that gene. You get two copies of the gene, one from your mom, one from your dad. They're missing one functional copy. They still make CCR5 because they've got one functional copy. That's about 10% of Northern Europeans are missing one. About 1% of Northern Europeans are missing both of them. They don't make a functioning CCR5, and they don't seem to get AIDS infected. So what He was saying is he's trying to come up with effectively a genetic vaccine for HIV by changing these babies' DNA so that it's very hard, if not impossible, for them to become HIV infected. That's not crazy in 15 or 20 years. It was crazy for him to do it now.
This is Stanford Legal, and today we're talking with Hank Greeley about genetic modifications and CRISPR. And can I just follow up a little bit, Hank, because you had been talking earlier about part of the problem here is there was nobody really to give consent for what was going on. I would assume that if the vast majority of people are producing CCR5, is that what it's
Yes.
called? That even if that increases your likelihood of becoming infected if you get exposed to the HIV virus, it presumably also has some evolutionary benefit, or at least might have some evolutionary benefit. So we don't know what happens if you lose it. Am I off base there?
Thank you, Pam, for supporting an argument I've long made that lawyers and scientists actually have a lot in common. That's a really good question. And it's not just that almost all humans have it. All mammals have it. It's likely that it's doing something. We don't really know what. We know that there are adults who seem to be healthy, Northern European adults who seem to be healthy who don't have it. There's some evidence that it makes you more susceptible to West Nile virus. There's some evidence it makes you get sicker when you get the flu, but we really don't know what it means not to have it. That's one of the two big risks to
Yeah, I mean, it's
these
kind of like
babies.
sickle cell, right? Which turned out to be very bad for some things, but also gave you more resistance to some forms of malaria.
Right, it's vaguely like sickle cell. The details are different, but the idea is right. Almost all genes do a bunch of different things, sometimes good, sometimes bad. So one risk to these babies is having their CCR5 knocked out will cause harms that we don't understand. Another risk is CRISPR is great. It's like the Model T. It's not the first gene editing, but it's the fastest, cheapest, best.
And does it come in any other color than black?
No. It's before the Model T. There were cars, but there weren't very many of them because people couldn't afford them. After the Model T, there were a bunch of them. That's what CRISPR does. It makes it so much easier and more affordable to make these kinds of changes. With the CCR5 gene, you could try to knock it out, but one thing we know is CRISPR's not perfect. And so what you worry about mainly are so-called off-target effects, where instead of going for AAA, CCC, GGG, TTT, ACGT, it instead hits AACA, CCC, and it goes someplace else, goes to a nearby address. Those off-target effects could do damage. And one of the really reprehensible things about what this guy did, he changed the embryos, and then before moving them into a woman's, or before having a technician move them into a woman's uterus for possible implantation, pregnancy, and birth, he checked them, he sequenced them through a procedure called Pre-Implantation Genetic Diagnosis, or PGD, that's been around for 28 years. And he discovered that one of the girls only had half of her cells changed, which means she basically has no protection against HIV, although she has the risks of having half of her cells changed. And the other girl had her cells, all of her cells changed, but they weren't changed, and all of her CCR5 genes changed, but they weren't changed in the way he wanted to change them, which was the way found in nature in 1% of the population. So there are risks both, maybe the CCR5 knockout's a bad thing, and maybe the CRISPR process that they used causes other bad things, and both of those risks were here for these babies. So the most important flaw in this experiment has nothing really to do with it being gene editing exactly. It's that the potential benefits to the research subjects and science were relatively trivial. The potential risks were hugely high. We don't let people do experiments where the risks are not counterbalanced by potential benefits. In 10 years or 15 years, when we know more about the safety both of CCR5 knockouts and of CRISPR, this might be a reasonable and acceptable experiment. Today, it's criminally reckless.
You know, Hank, I wanted to come back to where I started with, which was my desire to be taller, actually, not to have Scottish-looking hair.
Lifts. You need lifts.
Lifts.
Yeah.
Is this the kind of thing that in 10 years, in 20 years, you could use that for?
It depends on what trait you're looking for.
Yeah.
So unfortunately, as in law, biology is really complicated.
Mm-hmm.
Some things are simple, like blood type, A, B, O, A, B. Some things like height seem to be affected by more than 100 genes. So, and each gene has only a small effect, and we don't understand their interactions very well. So I could give you red hair much more easily because that's just one gene. I can fix your cystic fibrosis, fix your sickle cell anemia, fix your Huntington's disease. That's just one gene. You get into things that involve scores or hundreds of genes, we don't understand how they interact, and it's going to be very difficult to make all those changes. And unfortunately, the traits we are most interested in generally seem to involve lots and lots of genes.
Well, we'll be back with more from Hank Grilly talking about genetics and genetic modifications next on Stanford Legal on Sirius XM Insight 121. Welcome back to Stanford Legal, where we look at the cases, questions, conflicts, and legal stories that affect us all every day. I'm Pam Carlin, along with Joe Bankman. Hi, Joe.
Hi, Pam. Hank, before the break, we were talking about CRISPR in humans. Mm-hmm. But you believe that actually CRISPR might have more exciting or interesting or maybe even important implications for non-humans.
And terrifying. Its application in non-humans I think, has greater upside and much greater risks than its application to humans. Part of that is humans are not great lab animals. We have long generations, we lie and cheat, we have lawyers. Changing the human species one 30-year generation at a time is a really long run, and the things we care about most in humans are genetically complicated and hard to do. If you have a process that makes human babies and 10% of those babies end up dead or disabled, you should rot in hell. We care a lot about human babies. If, on the other hand, you have a process that makes 10% of mosquito babies dead or disabled, people applaud and say, why only 10%? So we will be able to do things in non-humans, animals, plants, bacteria, much, much faster and easier because it's not as risky, it's not as legally regulated for good reasons as doing work with human babies. We can do that for some really good things. The classic example is agriculture. I know GM foods are controversial for some, although not controversial at all among biologists. But let's say that so-called mythical global warming turns out actually to be real. And you're looking at moving the corn belt 200 miles further north. Well, if instead you can use CRISPR to change the genes of corn so that they can deal with hotter temperatures, a lot better. So a bunch of agricultural uses, things like turning algae into energy source uses. The one that's most interesting, though, involves mosquitoes. Mosquitoes kill more people than any other animal in the world. They got lions and tigers and bears beat by an order of 1 million. 450,000 people every year die from malaria. People don't give people malaria. Mosquitoes give people malaria, particularly species of a genus of mosquito called the Anopheles mosquito. Scientists are working on a way to make the Anopheles mosquito, to edit the genes of the Anopheles mosquito to make its immune system resist infection with the plasmodium, the thingy, the little bug that causes malaria. If the mosquito can't get malaria, people can't get malaria. And you save 450,000 lives a year. Now, Americans don't know about those 450,000 lives. They're almost all babies in Africa. But that's 450,000 people. That's more people than the U.S. lost in World War II.
So
and that's every single year.
The idea here would be you'd still have the mosquitoes because those are necessary for the bird life and the like, right? I mean,
maybe, right.
You don't necessarily want to get rid of the mosquitoes. What you want to do is get rid of them as being a vector for the transmission of malaria.
That's the most elegant version. People are also working on forms of CRISPR that make all the mosquitoes male, which crashes the mosquito population pretty quickly. So people are working on both reducing the numbers of mosquitoes, and people who don't like getting bitten by mosquitoes like that better, but that has, as you point out, the broader ecological and environmental
Yeah, I mean, that starts to look like what we were talking about before the break, which is, there might be a good reason for having mosquitoes. They just have this one quality that we'd like to change.
I take a chance on that, however.
So it depends on whether you're a bat or not, for example, that likes to eat mosquitoes. But there's another trick that you can apply to CRISPR called gene drive that pushes a change through a population really, really fast in a way that is amazing. So in theory, because CRISPR is so cheap, easy, democratic, an undergrad with a garage and $1,000 could, in theory, already be modifying the genes of mosquitoes. She could put them out there in Santa Clara County. She could find a time when Lake Lagunita actually has a little bit of water at the bottom of it, of the swampy lake log. Put them out there, and it's conceivable that within a couple of years, every mosquito in California could have this change, and we wouldn't even know it yet. Now,
are there any laws right now that tell you whether you can use CRISPR or not?
Yes, no, and maybe. It's a mess. In the mid-'80s, when GM food, genetically modified food, first became an issue, the Reagan administration pulled together a coordinated framework of laws affecting the Department of Agriculture, the FDA, and the EPA, none of which were written at a time when anybody had any idea what DNA was. And they said, we're going to apply these, we're going to stretch and edit these so they apply to DNA. It's really outdated at this point. Right now, if So EPA, at the end of the Obama administration, EPA and FDA made a deal that the Trump administration may be pulling back on. If you were reducing mosquito numbers, that was the EPA because it was a pesticide. If you were making mosquitoes safe from malaria, that was FDA because it was an animal veterinary drug. You were protecting these poor mosquitoes from getting malaria. The answer is we have no good framework, and the chances that Congress would pass good legislation on this issue, to say that they're slim and none, I think overestimates the possibilities.
This is Stanford Legal, and today we're talking with Hank Greeley about genetic modification and DNA. Joe?
Hank, I want to raise another subject that you've talked to the faculty about, which is using CRISPR to, how shall I put it, reinvent, reintroduce old species?
Yeah, de-extinction is the buzzword that it got. People wanted to call it resurrection, and I advised, no, that's a bad idea. Call it de-extinction. And that goes to, I think, a bigger issue, really. People look at the use of CRISPR in non-humans and say, well, agricultural uses, this public health use for mosquitoes, all these good uses. But like any tool, it can be used for good things, bad things, or things that are frivolous. And some of the bad things, smallpox is gone, except for a lab in Russia and the CDC. But there are a bunch of pox viruses, related pox viruses out there, like mousepox and monkeypox. We know the DNA sequence of smallpox. We know the DNA sequence of mousepox. A kid or a conspiracy with $1,000 in a lab and a couple of college courses could change mousepox into smallpox and kill hundreds of millions of people. Alternatively, you've got the ones in the middle. So I think the extinction is cool. I'd love to see a woolly mammoth, or my personal favorite would be a giant sloth. I want to see a giant sloth, maybe a saber-toothed cat.
Passenger pigeons.
Passenger pigeons. Actually, the thylacine, the Tasmanian tiger. So all you listeners, go to YouTube and put in Tasmanian tiger. You can see footage from the 30s of this really endearing dog-like creature that is extinct. Your grandparents could have seen it, but you can't. So you can do that. Or gardeners. People have tried for 200 years to breed blue roses. No one has succeeded. With CRISPR, blue roses should be easy. My personal favorite along these lines, and I think this is going to happen, and I think it's going to happen here in Silicon Valley. Some billionaire with a 12-year-old daughter is going to make her a unicorn for her birthday. Find the genes to make a single horn, put them into the embryo of a horse, and after probably a bunch of failed efforts, you get a unicorn. So people are going to play with this. Artists are going to use it. Scientists, except for this guy, usually dislike too much publicity and controversy. Artists love it. So I think CRISPR and our new biological tools in general are going to be used for purposes good, bad, ugly, indifferent, cool, weird, and playful.
God, that sounds like the dwarves.
I need 7, don't I?
Yeah.
So the woolly mammoth, how does CRISPR produce a woolly mammoth?
We don't know that it can, but here's the idea. We know mammoth's DNA. We've sequenced the DNA of about 20 different mammoths. They freeze in the permafrost. People have eaten mammoth stew. Tastes like chicken, but really chewy, bad chicken. We know their DNA. It turns out they're most closely related to Asian elephants. You make a cell line from Asian elephant cells, and bit by bit, you use CRISPR to change the Asian elephant DNA into the woolly mammoth DNA. You take that cell line and turn it into an embryo. You find a soon-to-be mama Asian elephant, and 22 months later, long gestation period, she's really confused when a hairy little calf pops out. There are people who are actually working on that. The passenger pigeon work is a little farther along. Basically, if you know the DNA sequence you want to get to, and you've got something that's close, you just start editing it.
So from curing malaria to de-extinguishing the woolly mammoth.
Yeah.
It's really a mouthful.
Yeah. And don't forget killing hundreds of millions of people through biological warfare.
I'm trying not to.
Yeah, well, let's
say
try
that
to. Yeah,
'cause
let's
I'm not sure
not
how
think
much
about
I
that.
trust every listener out there.
No, but I want to thank you, Hank, because as usual, having you on the show, we learned a huge amount, and Joe now has aspirations to be taller with red hair.
It's the freckles I'm looking forward to.
Yeah. And thanks to our listeners as well for joining us on Stanford Legal here on Sirius XM Insight 121.